Okay, so let's get right into it. We hear about clinical trials constantly, right? Especially anytime a new medicine or vaccine hits the headlines. But what are they really? Well, today, we're pulling back the curtain and unveiling the blueprint for how modern medicine is actually built.
I mean, think about it. You've probably wondered how we can be so sure a new pill or vaccine is actually safe and does what it's supposed to do. The answer isn't a guess or a hunch. It's a really powerful, systematic process designed to separate hope from hard facts. And that process is the clinical trial. So here's our game plan. We're going to start with the absolute basics. Then we'll look at the blueprint for how a trial is built. After that, we'll see that not all trials are the same. They have different goals and different designs. And finally, we'll talk about the single biggest enemy of good science, the fight against bias. All right, let's lay the foundation. Section 1, what is a clinical trial? When scientists and doctors use this term, what are they really talking about?
At its absolute core, it's pretty simple. A clinical trial is a tool. It's a structured experiment, It's not just an opinion, it's not an anecdote. It's a systematic, step-by-step way to figure out if a new drug, a new medical device, or even a new surgical technique actually works.
So why do we go through all this trouble? Well, sometimes it's about pure science, just trying to answer a really specific question to learn something new. But let's be real, there are also some very practical reasons. A company needs to run these trials to support a claim, basically to get the evidence they need for government approval, and of course to back up what they say when they market a new treatment.
You wouldn't build a skyscraper without a detailed blueprint, right? Well, a clinical trial is exactly the same. So let's look at that architectural plan, the one that takes a trial from just a cool idea all the way to a solid, measurable outcome. And it follows this really logical path. First, you start with a hypothesis, a core idea, something like, hey, I bet this new molecule can help with migraines. Step 2, you turn that idea into a very specific, answerable question. Then comes the intervention, that's the how. How much of the drug? How often do people take it? And finally, the outcome. You have to decide exactly what you're going to measure to see if it actually had an effect.
Now, here's something important. Not all trials are built to do the same job. Just like you'd use different tools for different tasks, you need different kinds of trials depending on the question you're trying to answer.
So, the biggest split is between watching and doing. In an observational study, researchers are kind of like wildlife photographers out in the field. They don't interfere, they just observe people and measure what happens naturally. On the other hand, you have an interventional study. This is where researchers get hands-on. They actually give an intervention, a new drug for instance, to one group and then compare their results to a control group to see what changes.
And we can break it down even further by purpose. The National Institutes of Health, or NIH, actually groups them into 5 main types. You've got prevention trials, trying to stop diseases before they even start. Screening and diagnostic trials are all about finding better, faster ways to detect and identify health problems. Treatment trials are probably the ones you hear about most, testing new medicines or surgeries. And then there are quality of life trials, which focus on making things more comfortable for people with chronic conditions.
Okay, so once you know your goal, you have to pick an architectural style, the actual structure of your trial. Let's dig into a few of the most common designs you'll see out there.
The most straightforward one is called a parallel design, and it's pretty much exactly what it sounds like. You get 2 or more groups of people, each group gets a different treatment, and they get them at the same time. The key thing is, you stay in your assigned group for the entire study.
The best way to picture this is like a swimming race. You have a pool of eligible participants, and you randomly assign them to different lanes. One lane gets treatment A, another gets treatment B. They swim in their own lane for the whole race, and at the end, you can directly compare who finished faster. It's clean, it's simple. But what if you could have the same person test both treatments? Well, that's the genius of a crossover design. In this setup, each person serves as their own control. They might start on treatment A for a while, then have a washout period. That's just time to let the first drug completely leave their system, and then they switch over, or cross over, to treatment B. Now this next one, the matched pair design, is a really clever way to make your comparison groups as similar as humanly possible. What you do is find 2 participants who are almost like twins on key factors, same age, same gender, same stage of a disease, and you put them together as a pair. Then you flip a coin. One person gets treatment A and the other gets treatment B. So you start with your whole group of volunteers, you screen them to make sure they're eligible, and then you start the matchmaking process. It's almost like a scientific dance-off. You pair up similar contestants, and then you see which move, or in this case, which treatment, gets the better score. It's a great way to control for a lot of variables right from the start. Okay,
Now we are getting to the absolute heart of what makes a clinical trial trustworthy. Because designing the trial is one thing, but protecting its integrity, well, that is a whole other battle. Every single researcher is in a constant fight to protect the truth from a very powerful and sneaky enemy.
So, what is it? What's this one thing that can creep in and completely undermine even the most perfectly designed study, potentially leading everyone to the wrong conclusion?
It's bias. Bias is any influence, conscious or unconscious, that can warp the results. A perfect example of this is the placebo effect. If a patient truly believes they're getting an amazing new drug, that belief alone can cause real, physical improvements. That's a powerful form of bias, and it absolutely has to be accounted for.
So how do we fight back? Well, scientists have a whole arsenal of weapons. We make sure the groups are as similar as possible from day one. We measure everyone's outcomes in the exact same standardized way. But the two heavy hitters, the real superheroes here, are randomization, assigning people to groups by pure chance, and blinding. Blinding is all about hiding who gets which treatment. Sometimes a trial is what we call open label. Everybody knows who's getting what. A step up from that is single blind, where the patient doesn't know if they're getting the real drug or a placebo. But the real gold standard is the double-blind trial. That's where neither the patient nor the doctor knows who's getting what. This is huge because it prevents the expectations of both people from influencing the results. And you can even go to triple blind, where the people analyzing the data don't know either.
But hang on, you might be thinking, how do you keep a trial blind if one treatment is a pill and the other is an injection? It'd be pretty obvious who's getting what, right? Well, scientists have this incredibly clever solution called the double dummy technique. Here's how it works. Group 1 gets the real pill and a fake placebo injection. Group 2 gets a fake placebo pill and the real injection. See, everyone gets a pill and a shot, so the blind is perfectly maintained. It's pretty brilliant.
So here's the big takeaway. The next time you see a headline that talks about a randomized, double-blind, placebo-controlled study, you'll know that's not just a bunch of fancy jargon. That's the signature of a high-quality trial. It's the sign that the researchers used the right blueprint to find and protect the truth. So knowing that, what's the first thing you're going to look for the next time you read about a big medical breakthrough?